Thyroid gland. The thyroid gland is a butterfly-shaped organ located in the base of your neck. It releases hormones that control metabolism. The thyroid hormones regulate vital functions of the body, including breathing, heart rate, central and peripheral nervous system, body weight, and muscle strength. Further functions of the thyroid hormone are increasing heart rate, increasing respiration, promoting growth and development of the brain during fetal life and for the first few years of postnatal life. Thyroid system. Hypothalamus secretes thyrotropin-releasing hormone which acts on anterior pituitary gland to release thyroid-stimulating hormone. Thyroid-stimulating hormone acts on thyroid gland to release T3 and T4 thyroid hormones. Increased concentrations of T3 and T4 in blood causes negative feedback mechanism to inhibit secretion of TSH from anterior pituitary gland and inhibit secretion of TRH from hypothalamus.
In the tissues of your neck is a small, unassuming organ that wields enormous power over your body. It's called the thyroid. Like the operations manager in a company, its role is to make sure that the cells in your body are working properly. It does that by using hormones to deliver messages to every single one of them. This high-ranking organ is made up of lobules that each contain smaller cells called follicles, which store the hormones the thyroid sends out into your blood. Two of the most important hormones it produces are thyroxine and triiodothyronine, or T3 and T4. As messengers, the hormone's job is to instruct every cell in the body when to consume oxygen and nutrients. That maintains the body's metabolism, the series of reactions our cells perform to provide us with energy. This hormonal notification from the thyroid gets the heart pumping more efficiently and makes our cells break down nutrients faster. When you need more energy, the thyroid helps by sending out hormones to increase metabolism. Ultimately, the thyroid allows our cells to use energy, grow, and reproduce. The thyroid is controlled by the pituitary gland, a hormonal gland deep in the brain that oversees the thyroid's tasks, making sure it knows when to send out its messengers. The pituitary's role is to sense if hormone levels in the blood are too low or too high, in which case it sends out instructions in the form of the thyroid-stimulating hormone. Even in this tightly controlled system, however, management sometimes slips up. Certain diseases, growths in the thyroid, or chemical imbalances in the body can confuse the organ and make it deaf to the pituitary's guiding commands. The first problem this causes is hyperthyroidism, which happens when the organ sends out too many hormones. That means the cells are overloaded with instructions to consume nutrients and oxygen. They become overactive as a result, meaning a person with hyperthyroidism experiences a higher metabolism, signaled by a faster heartbeat, constant hunger, and rapid weight loss. They also feel hot, sweaty, anxious, and find it difficult to sleep. The opposite problem is hypothyroidism, which happens when the thyroid sends out too few hormones, meaning the body's cells don't have as many messengers to guide them. In response, cells grow listless and metabolism slows. People with hypothyroidism see symptoms in weight gain, sluggishness, sensitivity to cold, swollen joints, and feeling low. Luckily, there are medical treatments that can help trigger the thyroid's activities again and bring the body back to a steady metabolic rate. For such a little organ, the thyroid wields an awful lot of power. But a healthy thyroid manages our cells so effectively that it can keep us running smoothly without us even noticing it's there.
Parathyroid gland. Parathyroid glands are small endocrine glands in the neck of humans and other tetrapods that produce parathyroid hormone. Humans usually have 4 parathyroid glands, variably located on the back of the thyroid gland. Functions of the parathormone include destruction of bone tissue and release of calcium to the bloodstream, activation of calcium absorption in intestines and reabsorption in the renal tubules. The sole purpose of the parathyroid glands is to control calcium within the blood in a very tight range between 8.5 and 10.5. Calcitonin and parathyroid hormone function.
Calcitonin is secreted by the thyroid gland. Parathyroid hormone is secreted by small masses of tissue located on the posterior side of the thyroid gland, called the parathyroids. Both hormones work to control blood calcium levels. Parathyroid hormone increases blood calcium levels, while calcitonin decreases blood calcium. Parathyroid works by stimulating bone cells called osteoclasts. These cells move calcium from the bones to the blood. Parathyroid hormone also increases calcium reabsorption in the kidney tubules, and it increases calcium absorption in the intestine by facilitating vitamin D production. Vitamin D is needed for calcium absorption. Calcitonin stimulates bone cells called osteoblasts. These cells move calcium from blood to bones. Calcitonin also inhibits calcium reabsorption in the kidneys, and it inhibits calcium absorption by the intestine. Both parathyroid hormone and calcitonin work to regulate blood calcium levels.
Adrenal glands. The adrenal glands are two glands that sit on top of your kidneys that are made up of two distinct parts, the adrenal cortex and the adrenal medulla. Adrenal hormones. Adrenal hormones can be classified according to the place of their secretion, such as secretions of adrenal medulla and secretions of adrenal cortex. Secretions of adrenal medulla are known as catecholamines, which includes hormone epinephrine and norepinephrine. Secretions of adrenal cortex can be divided according to the place of their secretion, where zona glomerulosa secretes mineralocorticoids, zona fasciculata secretes glucocorticoids, and zona reticularis secretes sex hormones. Functions of epinephrine and norepinephrine are: they affect vascular tone, inhibit GIT muscular tone, dilates bronchioles, and increase heart activity. They further involved in enhancing muscular activity and increasing blood glucose levels by lipolysis and gluconeogenesis. Mineralocorticoid hormones, which includes cortisol and aldosterone, takes part in control of mineral metabolism, increase sodium and chlorine reabsorption in renal tubules, and decrease potassium reabsorption in renal tubules. Glucocorticoid hormone can stimulate gluconeogenesis, decrease glucose utilization by cells, decrease extrahepatic tissue cellular protein mobilization of fat, and gives anti-inflammatory effects. Sex hormones mainly include androgens, progesterone, and estrogen. They are important when sex glands do not work during the childhood. Sex hormones function in development of genital organs and maintain metabolism and heart muscles, especially in women and during normal growth.
Adrenal gland. They are a pair of yellowish, flat, pyramid-like glands which lie over the upper end of the kidneys. They look like a cap on the kidney. Each gland has an outer yellow adrenal cortex, an inner reddish brown adrenal medulla. Adrenal medulla secretes two hormones called adrenaline or epinephrine and non-adrenaline or non-epinephrine. These are commonly called catecholamines. In conditions of physical and mental stress, such as fall in blood sugar levels, or when a person is injured or exposed to cold conditions or suffering with pain, it is also secreted during anger, fear, or grief. It helps the body to face these conditions. It is therefore called fight, fright, and flight. These hormones increase alertness, pupillary dilation, raising of hairs, sweating, etc. The hormone increases blood supply to heart and skeletal muscles. It constricts arterioles and blood supply to skin and gastrointestinal tract. The bronchioles dilate. There is increased rate of breathing, oxygenation, and heartbeat to meet any emergency. Catecholamines stimulate the breakdown of glycogen, resulting in increased concentration of glucose in blood. They also stimulate the breakdown of lipids and proteins. Adrenal cortex can be divided into three layers called zona reticularis, inner layer, zona fasciculata, middle layer, zona glomerulosa, outer layer. Adrenal cortex secretes many hormones called corticoids. A. Glucocorticoids involved in carbohydrate metabolism. Example, cortisol. B. Mineralocorticoids. Corticoids which regulate the balance of water and electrolytes in our body. Example, aldosterone. Glucocorticoids stimulate gluconeogenesis, lipolysis, and proteolysis and inhibit cellular uptake and utilization of amino acids. Cortisol is also involved in maintaining the cardiovascular system and kidney functions. It also produces anti-inflammatory reactions and suppresses the immune response. Cortisol stimulates RBC production. Aldosterone acts at renal tubules and stimulates reabsorption of Na plus and water excretion of K plus and PO4 minus. Thus, aldosterone helps in the maintenance of electrolytes, body fluid volume, osmotic pressure, and BP. Small amount of androgenic steroids are also secreted by adrenal cortex, which play a role in the growth of axial hair, pubic hair, and facial hair during puberty.
Pancreas. The pancreas is a glandular organ in the upper abdomen, but really it serves as two glands in one, a digestive exocrine gland and a hormone-producing endocrine gland. Functioning as an endocrine gland, the pancreas secretes the hormones insulin and glucagon to control blood sugar levels throughout the day. Glucagon and insulin are antagonistic hormones because their actions have opposite effects on the body. When the blood glucose level is high, insulin acts to lower it. When the blood glucose level is low, glucagon raises glucose levels. Thymus. The thymus is a specialized primary lymphoid organ of the immune system. Within the thymus, T cells or T lymphocytes mature. T cells are critical to the adaptive immune system, where the body adapts specially to foreign invaders. Thymus secretes thymosin, which stimulates the development of disease-fighting T cells. Gonadal hormones. Functions of the gonadal hormones. Progesterone involves in thickening the lining of the uterus each month. Estrogen stops FSH being produced so that only one egg matures in a cycle. It stimulates the pituitary gland to release the hormone LH. Prolactin produces milk in mammals. Relaxin relaxes the ligaments in the pelvis and softens and widens the cervix in preparation for the childbirth. Inhibin inhibits the secretion of FSH. Human chorionic gonadotrophin supports the ovarian corpus luteum, which in turn supports the endometrial lining and therefore maintains pregnancy. Human placental lactogen breaks down fats from the mother to provide fuel for the growing baby. Androgens. Androgens are crucial for male sexual and reproductive function. Gastrointestinal hormones. GI hormones are chemical messengers that implicated in many aspects of physiological functions of the gastrointestinal tract. They include gastrin, secretin, cholecystokinin, and gastric inhibitory peptide. Gastrin. Gastrin involves in secretion of HCl, increasing of intestinal movement, and releasing of pepsinogen. Secretin. Secretin involves in secretion of alkaline digestive proenzyme and inhibiting intestine motility. Cholecystokinin involves in secretion of proenzymes and bile. Gastric inhibitory peptide. It involves in insulin secretion and inhibit gastric secretions and motility. Kidney. Kidney produces three hormones: erythropoietin, calcitriol, and renin. Erythropoietin is a peptide hormone which regulates erythropoiesis. Renin regulates blood pressure and fluid balance in the body. The next hormone, calcitriol, stimulates the small intestine for protein synthesis, allowing absorption of calcium and phosphates. This ensures the availability of calcium and phosphate for bone growth. Calcitriol simultaneously activates osteoblasts to synthesize collagen. Heart. Atrial natriuretic peptide is a powerful vasodilator and a protein hormone which is secreted by heart cells. Atrial natriuretic peptide acts as a powerful vasodilator. It acts on outer adrenal cells to decrease aldosterone production, and also it acts on smooth muscle relaxation. Pineal gland. The pineal gland is located in the epithalamus near the center of the brain between the two hemispheres tucked in a groove where the two halves of the thalamus join. The hormone produced by the pineal gland is melatonin. Melatonin. Melatonin helps to maintain circadian rhythm and also it regulates reproductive hormones. Now let's take a look at the methods of hormonal assay. Hormones are present in extremely minute quantities in blood. Some concentrations are as low as 1 picogram. Therefore, it was difficult to measure these concentrations using chemical means. Hence, extremely sensitive tests were developed to measure these concentrations. This includes radioimmunoassay method and ELISA method. Now let's take a look at the radioimmunoassay method. Radioimmunoassay is a sensitive method for measuring very small amounts of a substance in the blood. This isotopic measuring method was developed in 1959 by two Americans, biophysicist Rosalind Yalow and physician Solomon A. Burson. Radioactive versions of a substance or isotopes of the substance are mixed with antibodies and inserted in a sample of the patient's blood. The same non-radioactive substance in the blood takes the place of the isotope in the antibodies, thus leaving the radioactive substance free. The amount of free isotope is then measured to see how much of the original substance was in the blood. How RIA works. As an example of how this technique works, let's apply it to insulin. To measure insulin, the first step is to mix known amounts of radioisotope tagged insulin and antibodies. These combine chemically. Next, a small amount of the patient's blood is added. The insulin contained in the blood displaces some of the tagged insulin. The free tagged insulin is then measured with isotope detectors and the patient's insulin level is calculated.
See the principle how the concentration of antigen is determined using radioimmunoassay. Now let us assume we have a serum sample and this serum sample contains A antigen. And now what we have to do? We have to determine the concentration of this A antigen present in the serum. Now if we want to determine the concentration of A antigen present in that sample, we must have three things with us. The first thing is the monoclonal antibody against the antigen whose concentration is to be determined. So here we must have the anti-A antibody with us. The second thing, the same antigen in radio labeled form. Means here we should have the A antigen in radio labeled form. And the third thing we should have is the A antigen in original form, that is unlabeled form. So these three things we must have with us if we want to determine the concentration of antigen using radioimmunoassay. The first step in radioimmunoassay is to code those monoclonal antibodies. So here the anti-A antibodies in the microtiter well. After this, after the coating the monoclonal antibody, then radio label antigens they are added to the well. But the concentration of radio label antigen is very high. It is very excess. So this concentration is able to saturate all the antigen binding sites of the antibody. So as you can see, all the antigen binding sites of the antibody they are saturated. So here we can say the radioactivity present on the antibodies, it is 100%. but there are some unbound radio-label antigen, and in the next step these unbound radio-label antigens, they are removed from the well with the help of washing. After this, a small fixed concentration of unlabeled antigen, for example say 1 nanogram, is added to the well. Now due to addition of unlabeled antigen to the well, there is creation of competition between radio-label antigen and unlabeled antigen, and this competition is for binding to the antigen binding site of the antibody. After this, the well is washed to remove any unbound antigen, whether it is radio-label antigen or unlabeled antigen, and then the radioactivity present in the well is determined. So as you can see, the number of radio-label antigen present on the antibodies, they are decreased, so ultimately radioactivity will also decrease, and this decrease in radioactivity is directly proportional to the concentration of unlabeled antigen added to the well. We have to record this data, how much unlabeled antigen was added to the well and to what extent the radioactivity was decreased due to this addition. Here we will consider that we have added 1 nanogram of the unlabeled antigen, and due to that addition the radioactivity remained in the well is say 90%. Now we have to keep repeating these steps, but just by increasing the concentration of unlabeled antigen. Now this will lead to the generation of the standard data, in which we will have the concentration of the unlabeled antigen added to the well and the respective decrease in the radioactivity. Using this data, we can draw a standard graph in which the concentration of antigen is plotted on the x-axis and the respective radioactivity, the decrease in the radioactivity, is plotted on the y-axis. And this graph can be used for the determination of concentration of the A antigen, as we have considered, which is present in the sample. After generation of this standard data, now again we have to repeat all these steps, but this time instead of adding unlabeled antigen in the third step, we have to add the sample. So as earlier, again we have to coat the monoclonal antibodies, again add the radio-label antigen at a concentration which will saturate all the antigen binding site. Again we have to wash the well, but this time we have to add this sample which contains the A antigen. Now we don't know what is the concentration of antigen present in the sample, but according to the concentration it will create the competition between the antigen present in the sample and the radio-label antigen, the same antigen in the radio-label form which is bound to the antibody. Now this competition will be proportional to the concentration, and according to the concentration of A antigen present in the sample, it will remove the radio-label antigen from the antibody. So ultimately the decrease in the radioactivity this time will be proportional to the concentration of A antigen present in the sample. So again, after addition of the sample, we have to wait to competition to occur, then we have to wash the well to remove any unbound antigen, and finally we have to record the radioactivity present in the well. If this value of radioactivity is extrapolated on that standard graph, now it can give us the value of concentration of antigen A present in the sample. For example, if the radioactivity remained in the well is 50%, now we can extrapolate this value on the graph and it can give us the concentration of A antigen present in the sample, that is 5 nanograms of the A antigen. So in this way The concentration of any antigen, a small antigen, can be determined present in the fluids like serum, etc.
Now let's take a look at enzyme-linked immunosorbent assay method. Enzyme-linked immunosorbent assay, also known as an enzyme immunoassay, is a biochemical technique used mainly in immunology to detect the presence of an antibody or an antigen in a sample.
The enzyme-linked immunosorbent assay, or ELISA, is an extremely sensitive test that is used to detect antibodies or specific antigens. The direct ELISA is a test for the presence of antigen. In this procedure, a known antibody is adsorbed to the inside of the well in a microtiter plate. After rinsing to remove excess antibody, the sample suspected of containing the antigen is added. Next, an enzyme-linked antibody that can react with the antigen is added. If antigen is present in the well, the enzyme-linked antibody binds to it and is retained. The colorless substrate for the enzyme is then added. Development of color indicates the presence of the antigen. In the indirect ELISA, antigen is added to the microtiter plate well, and the antigen attaches to the walls of the microtiter plate. After rinsing to remove excess antigen, the serum suspected of containing the antibodies is added. Enzyme-linked antibody capable of reacting with the constant region of other antibodies is then added, followed by addition of the colorless substrate. Development of color indicates the presence of the antibody being identified.
For this experiment, you will need a microtiter plate, phosphate buffered saline, antibody and antigen samples, a color-changing substrate solution, a 37-degree incubator, an adjustable micropipette and tips, and transfer pipettes. Step 1. Label the microtiter plate and transfer pipettes as noted in the product literature. Step 2. Add 100 microliters of the antigen solution to all of the wells. Step 3. Incubate for 5 minutes at room temperature. At this time, the antigens will non-specifically adhere to the plastic through hydrophobic and electrostatic interactions. Step 4. Remove all the liquid from the wells using a transfer pipette. Step 5. Wash the wells by adding approximately 8 drops of PBS buffer to each well. Remove all the PBS from each of the wells, taking care to prevent the buffer from spilling over into adjacent wells. Traditionally, following this step, the wells are blocked with a protein-containing buffer to prevent non-specific interactions between the antibody and the plastic wells. We have optimized this experiment to eliminate this step. Step 6. Add reagents as outlined in the product literature. Remember to use a clean micropipette tip for each reagent. Step 7. Incubate the plate for 15 minutes at 37 degrees Celsius. Step 8. Remove the liquid from the wells using the appropriately labeled transfer pipette. Step 9. Wash each well with fresh PBS. Remove the liquid using the transfer pipette designated for each sample. Step 10. Add 100 microliters of the secondary antibody to each of the wells. Step 11. Incubate for 15 minutes at 37 degrees Celsius. Step 12. While the samples are incubating, prepare the detection substrate as specified in the product literature. Step 13. Remove the secondary antibody solution from the wells using the transfer pipette designated for each sample. Wash each well once with fresh PBS buffer. Remove the liquid using the transfer pipette designated for each sample. Step 14. Add 100 microliters of the substrate solution to each well. Step 15. Incubate at 37 degrees Celsius for 5 minutes. Step 16. Examine your results. Differences between negative and positive samples will be obvious, with most positive samples turning brown in color. If color is not fully developed after 5 minutes, incubate at 37 degrees Celsius for a longer period of time.